Hello, my dear students. So let us start discussing the question paper. Question number 1 is which protein factor will come and bind to RNA polymerase and stop transcription എന്നാണ്. അതായത് stop transcription. Stop transcription. അപ്പോൾ you know that we have to focus on prokaryotes. As per our syllabus, we have to focus on prokaryotes. How will you define transcription? Transcription is the process of formation of RNA. Obviously, the next question is which RNA? Actually, it is any RNA. Transcription is the process of formation of rRNA, tRNA, mRNA. But we focus more on mRNA. And that happens in transcription unit. So, transcription occurs in transcription unit. So, if you can recall, there are 3 regions in transcription unit. There are 3 regions in transcription unit. This is your promoter. This is your structural gene. And this is your terminator. So you got promoter, you got structural gene, and you got terminator. We all know in prokaryotic transcription, initiation factor is sigma factor. Initiation factor is sigma factor. And termination factor is obviously rho factor. Termination factor is rho factor. So sigma factor is your initiation factor. Sigma factor is your initiation factor. And rho factor is your termination factor. So this is for initiation of transcription. This is for initiation of transcription. And this is for termination of transcription. In prokaryotes, eukaryote initiation factor, termination factor, നമുക്ക് പഠിക്കാൻ അങ്ങനെയില്ല. അപ്പോൾ ഇവിടെ ക്വസ്റ്റ്യൻ വന്നിരിക്കുന്നത് which protein factor will come and bind RNA polymerase and stop transcription near terminator? Termination of transcription is by termination factor near terminator. So answer is rho factor. So for question number 1, answer is 4. So rho factor is called termination factor in prokaryote, and sigma factor is called initiation factor in prokaryotes. With that, I come to question number 2. Question number 2 says match the following. Okay. In match the following, what is the concept? They have said coding strand, sigma factor, UTRs, template strand, and tailing. So coding strand. What do you mean by coding strand? We know that consider if this is your transcription unit. If this is your transcription unit, if this is promoter, this is structural gene, and if this is terminator. So this will be 3 prime, this will be 5 prime, this will be 5 prime, this will be 3 prime. And this strand, this is what is called as coding strand. And what is the direction of coding strand? 5 prime to 3 prime. So first 5 prime to 3 prime. So one is E. Second one is sigma factor. നമ്മൾ കുറച്ചു മുമ്പേ ഡിസ്കസ് ചെയ്തേ ഉള്ളൂ. That sigma factor is initiation factor. So second factor is the untranslated regions. So when you talk about untranslated region, എന്താണ് അതിൻ്റെ അർത്ഥം? Untranslated region എന്ന് പറയുമ്പോൾ പൊതുവെ 5' എൻഡിലും ആവാം, അല്ലെങ്കിൽ 3' എൻഡിലും ആവാം. പക്ഷേ കുറച്ചുകൂടി കൃത്യമായി നോക്കുകയാണെന്ന് വെച്ചാൽ ലുക്സ് ലൈക്ക് മാച്ച്, ഈ മാച്ചസ് വെച്ച് നോക്കുകയാണെന്ന് വെച്ചാൽ 3 ഈസ് A. So UTR it is 5' end. Ini template strand. Template strand is always 3', 5'. Template strand is always 3', 5'. So template, upper answer is B. Answer is B. And then finally tailing. Tailing is at 3' end. So C. So looks like E, D, A, B, C. Answer is 4. Yeah. So for question number 2, 4th is the answer. Yeah. With that I come to question number 3. When I go with question number 3, identify the correct sequence of steps in Hershey and Chase experiment. Hershey and Chase experiment. Hershey and Chase experiment was conducted in the year 1952. There are 3 stages in experiment: infection stage, blending stage, and centrifugation stage. Infection stage, blending stage, and centrifugation stage. And you know the radioisotope, they were used P32 in DNA and S35 in capsid. And you know capsid is otherwise called protein coat. So answer is infection, blending, and centrifugation. So infection, blending, and centrifugation, answer is 2. So for question number 3, 2 is the answer. Infection, blending, centrifugation. With that I come to question number 4. Which of the following is not correct? So which of the following, question number 4 is which of the following is not correct? Let us see. Option 1, the I gene codes for repressor of the lac-operon. This is correct. You know, in lac-operon, a regulator gene is represented by lac-I. And you know in lac-operon that lac-I stands for inhibitor. In lac-operon, I stands for inhibitor. And it is lac-I which will undergo transcription followed by translation, resulting in the formation of repressor or repressor protein. So this statement is correct. Let me go to the next statement. The Z gene code for beta-galactosidase, correct. Lac-Z, beta-galactosidase, lac-Y, permease, and lac-A, transacetylase, Scherian. So this statement is also correct. Lactose is the substrate for enzyme beta-galactosidase. Very, very important. Correct. You know what is the other name of lactose? You should know that lactose is otherwise known as beta-galactoside. Lactose is otherwise called beta-galactoside. Lactose, which is otherwise called beta-galactoside, is digested by the enzyme beta-galactosidase. So you should know that beta-galactosidase is otherwise called lactase. So it's a correct statement. The repressor of the operon is synthesized by the Z gene, 30. Z gene synthesizes beta-galactosidase. So this is a wrong statement. Answer is this one. So not correct, 4th one. Because the repressor of the operon is synthesized by lac-I, not by lac-Z. So for question number 4, 4 is the answer. With that I come to question number 5. So if you look at question number 5, which of the following is not correct about genetic code? If you go to page number 111 and 112, 111 towards last paragraph and page number 112, you can see genetic code chart. Second year biology NCERT. ഇവിടെ ചോദിച്ചിരിക്കുന്നത് നോട്ട് കറക്റ്റ് എന്നാണ്. ഫൈൻ. So here what is the condition? Genetic code is a triplet, that is correct. Triplet nature of genetic was proposed by George Gamow. Genetic code has to be triplet. The genetic code has to be triplet was proposed by George Gamow and not Francis Crick. Francis Crick proposed the double helical model structure of DNA as well as the proposed copying mechanism called semi-conservative nature. So this is not correct. Code is non-overlapping, yes. Code is non-ambiguous and universal, correct. So for question number 5, 2 is the answer. With that I come to question number 6. During the transformation experiment, that means this experiment is talking about Griffith's experiment. (1928). During the transformation experiment, which among the following conditions produced fatal pneumonia? What do you mean by fatal pneumonia? Pneumonia that killed the mice. Anything that kills is called fatal. Anything that kills is called fatal. So let us try to understand. This is question number 6. Question number 6. During the transformation experiment, which among the following condition produced fatal pneumonia in mice? Let us try to understand this. We know that R strain is avirulent, and we know S strain is virulent. So R strain is avirulent and S strain is virulent. Okay. So in the step 1 of Griffith's experiment, what happened? In step 1 of Griffith's experiment, S form was injected to the mice and mice died of pneumonia. In step 2, R form was injected to the mice lived. In step 3, heat killed S form. Heat killed S form was injected to the mice, but the mice survived. And in step 4, mixture of heat killed S form and leaving R form was injected to the mice and the mice died. So let us see. Live R strain injected into fatal pneumonia. R strain originally fatal pneumonia cause cheyillello. Heat killed S strain and live. Yeah, that is the answer. That is the answer. Mixture of heat killed S form and leaving R form. Mixture of heat killed S form and leaving R form. That's a heat killed S form, it will not cause. Heat killed S strain, no. So, mixture of heat killed S form and leaving R form can cause fatal pneumonia. As a result, the mice died. With that, I come to question number 7. So, for question number 6, 2 is the answer. Let me go to question number 7. What is happening in question number 7? Question number 7 says, the conclusion that we can make from the following diagrammatic representation of lac operon is. You know, in lac operon what happened? In lac operon, you got regulator gene, promoter gene, operator gene, structural genes. Regulator gene, that is represented by lac I, where I stands for inhibitor. Then it is lac Z producing beta galactosidase, lac Y producing permease, and lac K producing transcysteines. In absence of lactose, you know the inducer in lac operon is lactose or yellow lactose. And you know the other name of lactose is beta galactoside. The other name of lactose is beta galactoside. So if you look at this diagram, page number 117 NCERT, it is very clearly shown that the repressor protein is present at operator That means this lac-operon is in switch off condition. This switch off condition. So let us see what is happening. About this is switch off. RNA polymerase. So here, RNA polymerase is prevented from translation. No, RNA polymerase does not go for translation. We must understand, and you should be very clearly knowing that RNA polymerase is involved in transcription. Adilenandalo RNA polymerase. RNA undakunna enzyme. And the process of formation of RNA is called transcription, not translation. Appo aditha thetta. Chodyam enganeyanu vecha, the conclusion that we can make from the following diagrammatic representation. Okay. Transcription continues, impossible. Because here it is off condition. RNA polymerase is prevented from transcription? Yes. That is the answer. Why like that? Operator gene repressor protein ullodtholam kaalam. Operator gene repressor protein ullodtholam kaalam. RNA polymerase ne LAC-Z, LAC-Y, LAC-A ഒരിക്കലും transcribe ചെയ്യാൻ പറ്റില്ല. Transcribe ചെയ്യാൻ പറ്റില്ല. So one thing is sure. അതാണ് നടക്കുന്നത്. RNA polymerase LAC-Z, LAC-Y, LAC-A അതായത് സ്ട്രക്ചറൽ ജീൻസിനെ ട്രാൻസ്ക്രൈബ് ചെയ്യണമെങ്കിൽ ഇവിടെ ഈ റിപ്രസർ പ്രോട്ടീൻ ഓപ്രോണിൽ പാടില്ല. ഈ റിപ്രസർ പ്രോട്ടീൻ ലാക്കോപ്രോണിനോട് അറ്റാച്ച് ചെയ്യാൻ പാടില്ല. അപ്പോൾ സ്വാഭാവികമായിട്ട് ഇറ്റ് വിൽ ബ്ലോക്ക് RNA polymerase prevent from transcription of LAC-Z, LAC-Y, LAC. Translation continues? No. So basically if you look at this diagram, it is like that there is no activity taking place in LAC-Z, LAC-Y, LAC-A. So when there is no LAC-Z, LAC-Y, LAC-A transcription, naturally there is no translation, nothing. So for question number 7, 3 is the answer. With that I go to question number 8. Match the following. Okay, let us try to. So these are genetic code. This genetic code chart you should know very, very clearly. So we know that UUU is phenylalanine. A is equal to 5. So 3 is ruled out. Answer can be 1, 2 or 4. So what is UUU? UUU is phenylalanine. CCC is proline. So B is A. C, 5, 1, that's what. Proline, 1. Let us talk like that. So this will be 1, 2, 3, 4. It is automatically understood. AA, lysine, 4. And GGG is glycine, 2. So 5, 1, 4, 2. 5, 1, 4, 2, no. 5, 1, 4, 2, no. Answer is 4. 5, 1, 4, 2. So when we talk about match the following, so you must remember all the codons. Okay. So if you want to know the codons of serine is UCU, UCC, UCA, UCG, AGU and AGC. This is second column, first 4 codon. This is fourth column, third box, first 2 codon. This is how we are supposed to understand. This is how it works. So match the following. This is how it goes. With that, so for question number 8, 4 is the answer. With I come to question number 9. In transcription. In transcription. Capping at 5' end means. Now that is quite interesting question. What I want you to understand. If you go to page number 111, paragraph 2 and paragraph 3 of NCERT, you will come to know that split gene arrangement is probably ancient feature of genome. And you must know that split gene is present in eukaryotes, not in prokaryotes. That is why you have coding sequences and non-coding sequences. So what happens is immediately after transcription in case of eukaryotes, hnRNA, heterogeneous nuclear RNA or hnRNA is formed. And this hnRNA has coding sequences. Then what are the coding sequences of hnRNA called? Coding sequences of hnRNA are called exons. And non-coding sequences of hnRNA are called introns. So this hnRNA has to be converted to mRNA. And when you convert hnRNA to mRNA, there are steps. There are 3 steps: capping, tailing, splicing. Then the question is, what is capping? Capping is addition of 7-methyl guanosine triphosphate at 5' end of hnRNA. Then what is tailing? Addition of about 200 to 300 adenylate residues at 3' tail end of hnRNA. And then it is splicing. Splicing helps to remove introns and join exons to form much shorter mRNA. So whatever mRNA is formed after RNA processing or processing level, that is much shorter than the hnRNA that is produced after transcription. So tailing is addition of about 200 to 300 adenylate residues at 3' tail end of hnRNA. Capping is addition of 7-methyl guanosine triphosphate at 5' end of hnRNA after transcription inside nucleus in eukaryotes without exception and in template independent manner. That means capping occurs without the help of DNA. Okay. Tailing is also template independent manner. The splicing is also template independent manner. So if we go by all these things, you need to understand. It cannot be adenylate, so this is ruled out. A cap of methyl guanosine phosphate, it is not methyl guanosine phosphate, it is methyl guanosine triphosphate. Methyl guanosine mRNA, no. From hnRNA you form mRNA. hnRNA mRNA form ആയി വരിക. A cap of methyl guanosine triphosphate at 5' end of hnRNA? Correct. So for question number 9, 4 is the answer. For question number 9, 4. Very clearly, 4 is the answer. With that, I come to question number 10. Genetic code is said to be degenerate because. Now, what is the meaning of degenerate? Let me tell you. When you say degeneracy, what do you mean to say? One amino acid can be specified. One amino acid can be specified by more than one codons except methionine and tryptophan. Because you know that methionine and tryptophan, they don't show degeneracy. Then there is another word which is called unambiguous. Unambiguous, where a specific codon always codes and specifies particular specific amino acid only. So these are the two features that we broadly see. Now here it is asked that degeneracy. What is degeneracy? One amino acid can be specified by more than one codons. And there are only two amino acids that do not show degeneracy, which include methionine and tryptophan. So genetic code is said to be degenerate because codon generate quickly now. One amino acid is coded by more than one codon. That's it. So answer is 2. So for question number 10, answer is 2. One codon code for more than one amino acid? No, that feature is ambiguous, which does not exist. Genetic code is not ambiguous. Genetic code is unambiguous. Genetic code is unambiguous, and definitely genetic code is not ambiguous. Universal nature of genetic code does not mean the genetic code will degenerate. അപ്പോൾ വളരെ വ്യക്തമായി നമ്മൾ മനസ്സിലാക്കണം. Degeneracy എന്ന് പറയുമ്പോൾ where a given amino acid can be specified by more than one codon. ഒരു amino acidനെ ഒന്നിൽ കൂടുതൽ codon കൊണ്ട് code ചെയ്യാൻ പറ്റിയ, that is what is called as degeneracy. But when one particular codon codes and specifies a particular amino acid only, that is called as unambiguous. That's how you are supposed to understand. So for question number 10, 2 is the answer. With that I come to question number 11. Most abundant RNA is. This is previous year NEET question also. Most abundant RNA. You see, if you look at mRNA accounts for 3 to 5% of total RNA. ഒരു സെല്ലിൻ്റെ ഉള്ളിൽ 3 to 5% mRNA ആയിരിക്കും. So mRNA is 3 to 5%. tRNA is 15 to 20%. So you got mRNA which is 3 to 5%. You got tRNA which is 15 to 20%. And when you look at rRNA accounts for 70 to 80%. 70 to 80% is rRNA. And small nuclear RNA, that's okay. So now the question, of course, that is not more than a few percentage. So here it is most abundant RNA, definitely it is rRNA. rRNA is most abundant RNA. So for question number 11, 3 is the answer. With that, I come to question number 12. So when we look at question number 12, which one of the following is not a stop codon? You know, stop codons, you should know. Stop codons are otherwise also known as nonsense codons. Stop codons are otherwise also called as nonsense codons, which include UAA, which is also sometimes referred to as ochre. UAG, which is sometimes otherwise referred to as amber, and UGA, which is sometimes also referred to as opal. So there is ochre, amber, opal. So which of the following is not a stop codon? UA, definitely a stop codon. And what I want to tell you, that stop codon, there are some features that you should always remember. Stop codons do not code for any amino acids. They do not code for any amino acids. Second thing that you should know, they do not have, stop codons do not have anticodons. Stop codons do not have anticodons. And third thing you should know, they do not have tRNA. Stop codons do not have tRNA. So this is how you are supposed to understand. Right? So you need to understand this very clearly. So here stop codon UA, UGA, UAG, answer is 2. You know AUG is dual function codon. You should know this. AUG is dual function codon. AUG is dual function codon. It will act as start codon, and in between, if it comes, it will code for the amino acid. So question number 12, 2 is the answer. With that, I come to question number 13. The weight in the sense, the diameter. Diameter of DNA, you know it is 2 nanometer or 20 angstrom unit. Because this is the average diameter. So who decides the diameter of DNA? The diameter of DNA is decided by average bond length of hydrogen bonds. The diameter of DNA is decided by average bond length of hydrogen bonds. So naturally, let us see. 10 angstrom unit, never. 20 angstrom unit? Yes, that is the answer. 12 nanometer, no. 3.4 angstrom unit is average distance. Average distance between base pairs. Average distance between base pairs. So naturally for question number 13, answer is 2. Genetic code consists of, we know, the genetic code consists of 3 letters. GUG, AUG, GGG, GAG. That is how you go. So naturally what will happen? Genetic code consists of 3 letters. So for question number 14, 3 is the answer. That is how we need to understand this. Okay. With that I come to question number 15. With that I come to question number 15. Which one is wrong about genetic code? Non-overlapping, that is correct. Degenerate, it is correct. The genetic code must be written without any punctuations. Or punctuations പോലും ഇല്ലാതെയാണ് genetic code എഴുതേണ്ടത്. അത് ശരിയാണ്. Genetic code ambiguous ആണോ? അല്ല. Genetic code unambiguous ആണ്. Ambiguous എന്ന് പറഞ്ഞാൽ എന്താ? One codon code for many amino acid. Adana ambiguous. But it's not like that. One codon codes only for particular specific amino acid. Adana sheri. And this is correct, and this is what is called as unambiguous. With that, answer is 2. So for question number 15, answer is 2. With that, I come to question number 16. Inducer in lac-operon is lactose. You know, in lac-operon, lac itself stands for lactose. Lactose or L-lactose or beta-galactoside. You know, lactose is otherwise called as beta-galactoside. Lactose is otherwise called as beta-galactoside. Page number 117, paragraph 1, paragraph 2. Okay. So for question number 16, 1 is the answer. With that I come to question number 17. Introns are removed by the process of splicing. I was discussing a little bit before that there is capping, tailing, splicing, and splicing is removal of introns and joining exons to make much shorter RNA, which is mRNA. So splicing is carried out by spliceosomes or ribozyme proteins complex. So for question number 17, 4 is the answer. With that I come to question number 18. The part of transcription unit which is recognized by RNA polymerase for initiation. You know, for initiation, promoter is the region that is recognized by RNA polymerase with the help of sigma factor. And thereby sigma factor is called initiation factor. Sigma factor is therefore it is called initiation factor. So it is promoter. Promoter na thodangum, terminator vare povum, structural gene template strand transcribe cheyum. So question number 18, 3 is the answer. With that I come to question number 19. Question number 19, Mendel conducted hybridization experiment in the plant garden pea, Pisum sativum, where 2n is equal to 14. It's a very common question. So for question number 19, 3 is the answer. With that I come to question number 20. Question number 20, in the monohybrid genotypic ratio, for example, 1:2:1 is capital T is to capital T small t is to small t small t. We know that. F2 lele. So in the monohybrid genotypic ratio 1:2:1, numerical 2 stands for heterozygous dominant. Answer is 2. So for question number 20, answer is 2. അത് heterozygous dominant ആണ്. With that I come to question number 21. Mendel crossed the tall plant from F2 with the dwarf plant. This is testosterone. Unknown genotype. ഇതൊരു unknown dominant genotype ആണ്. Unknown dominant genotype ആണ്. അതിനെ recessive parent കൊണ്ടാണ് ക്രോസ് ചെയ്തെടുക്കണമെങ്കിൽ recessive parentനെ കൊണ്ട് ക്രോസ് ചെയ്യണമെങ്കിൽ that is what is called as testosterone. Either of the parent is back cross. But when you cross it with dwarf plant means recessive, so it is testosterone. And testosterone significance is to find out the unknown genotype of the dominant parent. Our dominant ഏതാണെന്ന് അറിയില്ല, ഹോമോസൈഗസ് ആണോ ഹെട്രോസൈഗസ് ആണോ എന്ന് കണ്ടുപിടിക്കാൻ വേണ്ടി ചെയ്യുന്ന ക്രോസാണ് testosterone. So testosterone, the significance of testosterone is to find out whether that unknown dominant genotype is homozygous or heterozygous. So testosterone is a type of back cross, but all back crosses are not testosterone. So for question number 21, 1 is the answer. With that I go to question number 22. Question number 22, flower color of snapdragon or dog flower, which means Antherinum species. Which means Antherinum species. Incomplete dominance. We know that. Complete dominance, IAI example. Multiple alleles, blood grouping. Codominance, AB blood group. അങ്ങനെയാണ് നമ്മൾ എക്സാമ്പിൾ ഇതിൻ്റെ പഠിക്കുന്നത്. അപ്പോൾ ഫ്ലവർ കളർ ഓഫ് സ്നാപ്ഡ്രാഗൺ ഓർ ഡോഗ് ഫ്ലവർ ഓർ ആന്ത്രനം സ്പീഷീസ് ആൻഡ് ഫ്ലവർ കളർ ഓഫ് മിറാബിലിസ് ജലാപ്പ വിച്ച് ഇസ് അതർവൈസ് കോൾഡ് ഈവനിങ് പ്രിംറോസ് ഈസ് ക്ലാസിക്കൽ എക്സാമ്പിൾ ഓഫ് incomplete dominance. So for question number 22 answer is 2. With that I come to question number 23. Question number 23 says which among the following blood group is an example of codominance? നമ്മൾ ഇപ്പോൾ ഡിസ്കസ് ചെയ്തേ ഉള്ളൂ. AB group only. How do you say that? You see, when you write IAI, A is a sugar that protrudes in RBC. A is a sugar that protrudes in RBC. B is the sugar that is present in B. So A is dominant over O, B is dominant over O, but the RBC, if it contains both the sugar A and B, codominant, both the genes are expressed. So AB blood group is a classical example of codominance. Adu matralla heterozygous HbA, HbS genotype of sickle cell anemia is also an example of codominance. With that, I come to question number 24. If the blood group of one parent is B group and the offspring is of O, which among the following blood group is not possible for second parent? So, നമുക്ക് heterozygous case എടുക്കാം. IB I ആണ്, കുട്ടി I ആണ്. അപ്പോൾ ഒരു കാര്യം ഉറപ്പാ, which among the following is not possible? AB ഒരിക്കലും പോസിബിൾ അല്ല. കാരണം അത് AB ആണെങ്കിൽ അതിൻ്റെ ക്രോസ് എങ്ങനെ വരിക? AB ആണെങ്കിൽ ഇതിൻ്റെ ക്രോസ് എങ്ങനെ വരിക? ഇത് P1, ഇതെന്ന് G1, IB encircled, I encircled, IA and IB it is encircled. So you got like this, paternal side, maternal side, this is IB, IA, IB. So this is IA, IB, this is IAI, this is IB, IB, this is IBI. ഒരിക്കലും O ബ്ലഡ് ഗ്രൂപ്പ് ഇവിടെ വരില്ല. ഇപ്പോൾ B ആണെച്ചാൽ ഹെട്രോസൈഗസ് എടുക്കാമല്ലോ. B ആൻഡ് O ആണെങ്കിൽ എന്തായാലും B ആൻഡ് O ആണെങ്കിൽ കുട്ടി O ആവാം, A ആൻഡ് O ആണെങ്കിലും കുട്ടി O ആവാം. AB ആണ്. സോ ആൻസർ ഈസ് 3. AB വന്നാൽ ഇവിടെ O ബ്ലഡ് ഗ്രൂപ്പ് വരുന്നുണ്ടോ? ഇല്ല. അപ്പോൾ ദിസ് ക്യാൻ നോട്ട് ബി ദ ബ്ലഡ് ഗ്രൂപ്പ് ഓഫ് വാട്ട് ഈസ് സോ കോൾഡ് ആസ് the second parent. So for question number 24, 3 is the answer. With that I come to question number 25. In case of pattern of inheritance, the phenotype reflects the contribution of each allele. Additive എന്ന് വെച്ചാൽ polygenic inheritance. അപ്പോൾ ഇതിൽ polygenic inheritance-ന്റെ example human skin color, human eye color, human skin color, human eye color, and the height of the humans. ഈ 3 എക്സാമ്പിൾ പോളിജീനിക്കിൻ്റെ എക്സാമ്പിൾസ് ആണ്. ഒന്ന് ഹൈറ്റ്, 2 സ്കിൻ കളർ, 3 ഐ കളർ. ABO ബ്ലഡ് ഗ്രൂപ്പിംഗ് മൾട്ടിപ്പിൾ അല്ലേഴ്സിൻ്റെ എക്സാമ്പിളാണ്. അപ്പോൾ ഹ്യൂമൻ സ്കിൻ കളർ ക്യാപിറ്റൽ A, ക്യാപിറ്റൽ A, ക്യാപിറ്റൽ B, ക്യാപിറ്റൽ B, ക്യാപിറ്റൽ C, ക്യാപിറ്റൽ C ആണെങ്കിൽ ഇത് ഡാർക്കസ്റ്റ് സ്കിൻ കളർ ആയിരിക്കും. സ്മോൾ a, സ്മോൾ a, സ്മോൾ b, സ്മോൾ b, സ്മോൾ c, സ്മോൾ c ആണെന്ന് വെച്ചാൽ അത് ലൈറ്റസ്റ്റ് സ്കിൻ കളർ ആയിരിക്കും. ക്യാപിറ്റൽ A, സ്മോൾ a, ക്യാപിറ്റൽ B, സ്മോൾ b, ക്യാപിറ്റൽ C, സ്മോൾ c. അപ്പോൾ ഓരോ അല്ലിലും ആ സ്കിൻ കളറിന് കോൺട്രിബ്യൂട്ട് ചെയ്യുന്നുണ്ട്. ഈ ചെല്ലിൽ കോൺട്രിബ്യൂട്ട്സ് ഫോർ ദാറ്റ് പെർട്ടിക്കുലർ കളർ. ഓരോ അല്ലിലും ആ കളറിന് കോൺട്രിബ്യൂട്ട് ചെയ്യുന്നുണ്ട്, ആഡിറ്റീവ് ഉണ്ട്. അപ്പോൾ അത് പോളിജീനിക് ഇൻഹെറിറ്റൻസ്. സോ ഫോർ 25(1) is the answer. With that, I come to question number 26. Alleles of one gene is always present in. We all know that alleles of one gene is always present in the same locus of homologous chromosomes. Same locus of homologous. Answer is 3. അപ്പോൾ ഇതാണ് ഒരു ക്രോമോസോം, ഇതാണ് ക്രോമോസോം, ഇത് ഹോമോലോഗസ് പെയർ ആണ്. ഇവിടെയാണ് ടി ആണെങ്കിൽ ഇവിടെ ആയിരിക്കും ടി. ഹോമോലോഗസ് പെയർ കണ്ട ഒരേപോലുള്ള. സോ ഹോമോലോഗസ് പെയറിൻ്റെ ഒരേ ലോക്കസിലായിരിക്കും. ഇതാ കണ്ടോ, ഒരേ ലോക്കസിലായിരിക്കും. അപ്പോൾ ഇതാണ് ഓരോ ക്രോമോസോമിൻ്റെ ഓരോ ലോക്കസിലായിരിക്കും. സോ സെയിം ലോക്കസ് ഓഫ് ഹോമോലോഗസ് ക്രോമോസോം. 26(3) is the answer. With that, I come to question number 27. Question number 27. Column 1, column 2. So let us see. Mendel, 1856. Mendel is ഇങ്ങനെ ബെസ്റ്റ് ഔട്ട് ഓഫ് 3 എടുക്കുമ്പോൾ Mendel started his experiment in the year 1856. Langdon Down discovered Down syndrome 1866. So B is 4. Right? Then C. Walter Sutton 1902. Eric von Schermach during 1900. These are rediscoverers of Mendelism. So D will be 2. 3, 4, 1, 2. Answer is 3. So for question number 27, 3 is the answer. With that, so this is how it will be. 1902, Sutton chromosomal theory of inheritance. 1900, rediscovery of Mendelism. 1856, Mendel started with the experiment, and 1866, Langdon Down syndrome. So 27, 3 is the answer. With that, I come to 28. Question number 28 says, which among the following is wild type? Wild type means dominant type. You know dominant type is represented by symbol plus. Wild type. Appo plus, idana answer. 1. Small y, small w without plus symbol recessive ആണ്. Small y, small w with plus symbol dominant ആണ്. അതാണ് wild. ഇത് രണ്ടും കണ്ടോ, റിസസീവ് ആണ്. ഇത് ഡോമിനൻ്റ് ആണ്, പക്ഷേ ഇത് റിസസീവ് ആണ്. ഇത് രണ്ടും റിസസീവ് ആണ്. അപ്പോൾ ഒന്നില്ലെങ്കിൽ വൈൽഡ് ടൈപ്പ്, അല്ലെങ്കിൽ ഡോമിനൻ്റ് ടൈപ്പ്, അല്ലെങ്കിൽ നമ്മൾ പ്ലസ് സിമ്പിൾ ഇടും. അപ്പോൾ വളരെ വ്യക്തമായിട്ട് 28 ആൻസർ ഈസ് 1. വിത്ത് ദാറ്റ് ഐ കം ടു ക്വസ്റ്റ്യൻ നമ്പർ 29. Chromosomal basis of sex determination by Henking in the year 1891 on the basis of observation made on insects, that is grasshopper. He was studying grasshopper, and you know grasshopper has autosome plus XO and autosome plus XX. Autosome plus XO and autosome plus XX. So this will be male, this will be female. So that is how. So male produces 2 types of sperm. So grasshopper, it has XO type of. So sperm with X and without X. That is how. So answer is insect. So for question number 29, 2 is the answer. With that, I come to question number 30. The 2 sexes in the population are having difference in chromosome number. Which respective number of sets of chromosome in a cell is found is? So that is the question. Question number 30. The 2 sexes in the population are having difference in chromosome with number of sets. Means honeybee. honeybee male is haploid with 16 and female is diploid with 32. ഇതിൽ ഒരു സെറ്റ് കുറവല്ലേ? With respect to the number of sets of chromosome in a cell found, അതാണ് പറഞ്ഞത്. Honeybee, cockroach XXXO, grasshopper XXXO സെറ്റ് അല്ല, ഒരു ക്രോമോസോമേ കുറവുള്ളൂ. സോ ആൻസർ ഈസ് honeybee. ഇതിലാണ് ഒരു സെറ്റ് കുറവ്. മെയിലിൽ ഹാപ്ലോയിഡ് ആണ്, ഫീമെയിൽ ഡിപ്ലോയിഡ് ആണ്. സോ ക്വസ്റ്റ്യൻ നമ്പർ 33 is the answer. With that I come to question number 31. Question number 31, the frame shift mutation. Question number 31, the frame shift mutation. Too closely linked gene frame shift mutation is alpha thalassemia. Chromosome 16 ആണെങ്കിൽ alpha thalassemia. Chromosome 11 ആണെങ്കിൽ beta thalassemia. Right? Hemophilia is X-linked. ഇത് autosomal ആണ്. So answer 16th ആയതുകൊണ്ട് alpha. So question number for 31. Question number 31. Chromosome 16th frame shift mutation ആണെങ്കിൽ നമുക്കറിയാം alpha thalassemia ആണ്. Chromosome 11 ആണെങ്കിലാണ് beta thalassemia. So answer is 1. With that I come to question number 32. Monosomy. You know that aneuploidy means addition or deletion of one or more chromosomes. ഒന്നോ ഇതാണ്. അപ്പോൾ aneuploidy ട്രൈസോമി ആണെങ്കിൽ ഒന്ന് ആഡ് ചെയ്യും. അപ്പോൾ ഇതിനെയാണ് നമ്മൾ 2n+1 എന്ന് പറയുക. നള്ളിസോമി ആണെങ്കിൽ അതിനെ നമ്മൾ 2n-2 എന്ന് പറയും. അല്ലെങ്കിൽ കുറച്ചും കൂടി സീക്വൻസിൽ എഴുതുകയാണെന്ന് വെച്ചാൽ ടെട്രാസോമി എന്ന് എഴുതാം. അപ്പോൾ ഓർത്ത് വയ്ക്കാൻ എളുപ്പമായിരിക്കും. ടെട്രാസോമി ആണെങ്കിൽ അത് 2n+2 ആണ്. മോണോസോമി ആണെങ്കിൽ അത് 2n-1 ആണ്. നള്ളിസോമി ആണെങ്കിൽ 2n-2 ആണ്. ഡൗൺ സിൻഡ്രോം ട്രൈസോമിയാണ്, 2n+1 ട്രൈസോമിയാണ്. ക്ലിൻഫിൽട്ടറും ട്രൈസോമിയാണ്, 44+XXY ആണ്. ഇത് 45+XX or 45+XY. ടർണർ സിൻഡ്രോം മോണോസോമിയാണ്. സോ NCERT പ്രകാരം മോണോസോമി എന്ന് വെച്ചാൽ ടർണർ സിൻഡ്രോം 44+X. ഇതാണ്. So for 32, 3 is the answer. With that, I come to question number 33. Which of the following is incorrect regarding origin of life? So if you see question number 33, oxygen combined with carbon dioxide and water to form methane and ammonia. തെറ്റ്. It is carbon dioxide. It is not carbon dioxide and water. You understand that? So about 2001, that is the first form of life slowly went through that. That is correct. First cellular form of life did not. Yeah, that's correct. So oxygen combined with methane and ammonia to form carbon dioxide, and so that is how it is. So this is a wrong statement. So for question number 33, 4 is the answer. With that I come to question number 34. Giant molecule present in first non-cellular form of life were RNA, protein, polysaccharide. Answer is 3. So for question number 34, it is a direct question. Answer is 3. Question number 34, answer is 3. RNA, protein, polysaccharide, giant molecules. Which one of the following is true regarding with embryological support for evolution by Ernst Haeckel? It says all vertebrate embryos are similar during early stages. All vertebrate embryos have a row of vestigial gill slit, but it is functional. Yeah, that is how. In NCERT, as per NCERT, we know Ernst Haeckel proposed ontogeny recapitulates phylogeny. An organism, when it is growing embryologically, it will pass through certain stages in which it will resemble the ancestral form. So this is correct one. With that I come to question number 36. Which one of the following is very least useful to determine. Close relationship. Analogous organs. Analogous organs, they develop among unrelated forms of related species. Wings of the bird and wings of insect. Convergent evolution, no common ancestry. Divergent evolution നമുക്ക് മനസ്സിലാവും, vestigial എന്നും മനസ്സിലാവും, homologous എന്നും മനസ്സിലാവും. Analogous organs common ancestry അല്ല. So for 36, 4 is the answer. With that I come to question number 37. Question number 37 says, branching descent and natural selection. ഇത് ടെക്സ്റ്റ് ബുക്കിൽ ബോൾഡായി കൊടുത്തിട്ടുമുണ്ട്. ഡയറക്റ്റ് ക്വസ്റ്റ്യൻ ആണ്. So for question number 37, 3 is the answer. With that I come to question number 38. According to De Vries, the mechanism of evolution is, mechanism of evolution is due to single step large mutation, and that is saltation. Single step large mutation, saltation. So question number 38 2 is the answer, direct question. With that I come to question number 39. Match the following. If you look at match the following, let us see. A 4. So A is 4. B 321. So 4 1. C, invertebrates were formed and active 500 million years ago. So 5. A coelacanth fish caught in South Africa 1938. 2. And fish like reptiles 200 million years. 3. So E is 3. 4 1 5 2 3. 4 1 5 2 3. Answer is 1. So for question number 39, answer is 1. With that I come to question number 40. The skull, adult human skull. That's what it's very clearly mentioned in NCERT. Question number 40. The skull of baby chimpanzee is more like that of adult human skull. Direct question. There is comparative skulls of baby chimpanzee, adult human, and given. Below that, in that diagram paragraph, it is written. So for question number 42 is the answer. Question number 41. Question number 41 says, pisciculture means fishes. Direct question. Apiculture, ornithology, that's okay. Pisciculture, fish. So for question number 41, 4 is the answer. Question number 42. Question number 42. Mating of superior males of one breed with superior female of another breed, that is crossbreeding. So answer is 4. Direct question. With that I come to question number 43. Hisardale, very, very important. Hisardale is a new breed of sheep. Direct question it is. Right, very simple direct question. With that I come to question number 44. Animal husbandry deals with techniques of artificial insemination cross, breeding and raising of livestock. That's it. That's what is animal husbandry. With that I come to question number 45. Question number 45, what it says. Okay. Question number 45. Semi-dwarf variety of rice introduced to India. IR8. So, one is B. If one is B, so this has to be answer. So, let us see. High yield wheat variety. It is Sonalika. So, answer is 2. Direct question. Question number 45, 2. And what is Himagiri? Himagiri, no, not Himagiri. Dwarf variety of rice developed in India. Jaya. Tropical canes of South India, thicker stem. D. So for question number 45, 2 is the answer. With that I come to question number 46. Among the following, which is bacterial disease? We know red rot of sugarcane, late blight of potato. That is fungal. Black rot of crucifer is bacterial disease. So answer is 4. With that I come to question number 47. Pusa komal. That is the question. Pusa komal is cowpea that is resistant to bacterial blight. In strategies, that box is given. We have to memorize. So it is a direct question. Pusa komal is cowpea resistant to bacterial blight. So with that, I come to question number 48. Question number 48, which of the following is correct? Okay, this is 48. So for question number 47, 2 is the answer. Question number 48. Question number 48. Spirulina is food rich in protein. Correct. It is a single cell protein. Atlas 66. Protein, high protein content. Hemagiri disease. Brassica? No. Hemagiri is wheat. So this is incorrect. Carrot and pumpkin vitamin A correct. So question number 48, 3 is the answer. With that, I come to question number 49. What is correct about single cell protein? It is the biomass of microbe. Correct. It include only bacteria? No. Bacteria, yeast. It contain only protein and minerals? No. Other nutrients also. Toxic microorganism we also used? No. So 49, 1 is the answer. With that, I come to last question. Question number 50. Part of a plant material which is used to initiate tissue culture is explant. So answer is 3. Explant is a part of the plant that is excised and used. In tissue culture. And you know this explant contain totipotent cells. And these totipotent cells are the cells which will help to generate entire plant from the parent plant. So that is how totipotency plays a major role when we talk about explant. And you know, tissue culture is raising the plant in vitro. Under proper nutrient condition, proper medium is given. And you raise the complete plant. That is what is called as tissue culture. Oxygen is growth factors are given. You with agar or without agar, suspension culture, or it can be callus culture. So explant, clone is genetically similar cell. Clone, whenever you say clone is genetically similar cell. Soma clonal cells are also genetically similar cell. Callus is mass of undifferentiated cells from which all these tissue culture things can be done. So I hope the exam was good. Keep learning, keep solving questions. God bless you. Thank you.